Scientist and former Vice Chancellor, University of Delhi South Campus, India, and also Dr. Bauju Gu, Research Plant Pathologist, USDA Agriculture Research Service, University of Georgia, USA, to come on the dais, and please welcome them to co-chair this session.
I see people slipping in very slowly after a very good lunch. Welcome to the third session of the day. Just like in the morning, we had some wonderful talks. We are looking forward to listening to the speakers of this session. With this ticking clock at the top, the idea of chairing anybody has become redundant. In fact, the clock can also tell whether any questions can be asked or not. That will be, I hope, the next development in managing conferences. My colleague here, Dr. Gao, I'm going to pass on the baton to him to run the show because I may have to slip out after 2-3 talks. And secondly, I would like to sit there and so I'm able to better see the slides. So over to you, Dr. Gao. It's your...
Yeah, thank you. You can see the 10 years, how the 10 years can make people, hopefully I cannot back to 10 years ago. But for science, the infinite, the 10 years really short. But the accomplishment and the contribution, we will remember forever. Appreciate Rajiv and to organize this conference, and certainly we all here to witness the accomplishment and the contribution of CEG, for last 10 years. Hopefully, everybody here we can together to give us our best wishes for next 10 years. We'll see how this CEGSB going to contribute to science. The bottom line is to contribute to the farmers. And my name is Bauju Guo here in the USDA and the UGA. I'm plant pathologist, mainly working on 2 crops, the peanuts and the corn. One thing for those 2 crops common is that food safety, aspergillus, aflatoxin, contamination. That's the one thing we are in common to, hopefully we can solve that problem to help the farmers and the consumers to have safe food. And with that said, we have our first speaker. It's Christian Jung from Germany. Our speaker here. It's all yours.
Thanks. So, good afternoon. I realize that I think I'm the first and maybe only speaker who's not from a big organization, as you heard before, but from a small institute from a university in northern Germany, as you can see, the University of Kiel. And instead, I'm very grateful to Rajiv that he gives me the opportunity to share some of our thoughts about flowering time regulation with you. Second, I will not talk much about genomics, but I will talk about how we use genomes and whole genome sequences to clone genes. And we'll talk more about genes and sequences and how we can exploit sequence information for breeding. The third is, unfortunately, is a crop you're not very familiar with, or most of you have heard for the first time. But the ideas and the strategies I will propose are, in general, are applicable to all vegetative crops. There are numerous. And this is linked to the phases of a flowering plant. So there are actually two major phases. After the juvenile phase, there's the vegetative phase. And you know that there are many crops where vegetative parts of the plant are harvested, like leaves or roots. And if you want to harvest these parts, the plant must not flower. So preventing flowering or floral transition is essential. And other plants, the majority of course, needs floral transition, otherwise you cannot harvest reproductive organs like seeds, fruits, tubers, flowers. So it's very important to understand floral transition, which is triggered by a number of genes in concert with environmental cues, like cold, temperature, or day length. And in the next minutes, I will show you how we work on a crop, understand floral transition, and how to exploit sequence information for improving this crop. Now the life cycle, as I said, is depending on day length and on cold. And this is true for crops from temperate climates, from northern climates like where I'm from, Central Europe, North America, Northern Asia. So these are all long day crops. And in the long day, they more or less quickly make floral transition and turn to the reproductive phase, like spring wheat and others. If you, and this on the right, let it grow under short days, it reacts in a way that it's not able to make floral transition. Instead, it makes a huge vegetative mass, but sets flowers and seeds, not flowers and seeds, or very late, which can be positive if you're interested in a vegetative crop, but it's negative if you're interested in seeds or others. So day length is an important trigger. A biennial crop, this was the annual, so finishing life cycle within the first year. A biennial crop, a plant in general, has a vernalization requirement. So in the first year, under long day conditions, it makes a huge vegetative mass, which in terms of a root crop is favorable. If you expose it to cold temperatures, which is shown on the right, or that is winter in northern climates, it starts in the next year, it starts flowering, like a winter crop, like winter wheat, barley. And this must be avoided for a root crop. So root crops or other vegetative crops face a problem. This is really a major problem worldwide. When crops, unfortunately, face cold temperatures due to early sowing, and this is an important aim in crop production for vegetables, for instance, cabbage production. Farmer wants to be early in the market, so he has to sow very early in the year, and what can happen in northern climates, cold temperatures are coming. And then it happens what you can see here on the left, the plant starts flowering and the crop is lost. On the other hand, we are interested in a crop which after winter, as you see on the right, after winter is not able to flower, but makes a huge and even more vegetative mass, which is a winter crop for a biennial vegetative crop. And I will show you some examples how to use this. Now, floral transition has been studied mostly in Arabidopsis. This is a simple picture of floral transition in Arabidopsis, about 150 genes we know today. I will not bore you with all the details. Today I will talk about orthologs of three major genes, the constans gene, which is a major gene from the photoperiod pathway, FT, the flowering locus T, which encodes the famous florigen, which is produced in the leaf and transported to the apical meristem, and PR7, where we found the homologin beet. And there are many other genes I will not talk about today. So the crop I'm talking about is beet, as a typical representative of vegetative crops. This is an old map where we have mapped candidate genes. And what we know since many years is that many of these genes from Arabidopsis are highly conserved and found in other species, even non-related, even in monocot species. And we look for sequence conservation, mapped floral transition genes from Arabidopsis, so the beet counterparts on the beet map. And in the next minutes, I will talk about 4 maps, 4 major genes, FT1 and FT2. These are 2 functional orthologs of the FT gene of Arabidopsis. Interestingly, one is a floral activator, FT2, but the other has an opposite function. And this is a very nice example of neo-functionalization during evolution, that a gene which in one species is a floral activator, in the other, during evolution, turned into a floral repressor. The next is B. This is a new gene we cloned from beet, and B2, another gene which recently cloned by MEP is cloning. And finally, briefly talk about a QTL for never bolting called BR1. Now, BTC1 is the gene which encodes the phenotypic variation at the bolting locus B. By the way, a very old locus, even proposed before Mendelian rules have been detected by German breeders as a genetic factor, a simple genetic factor. And 5 years ago, we could clone these genes from its position on chromosome 2. It's a transcription factor, as most of the genes you saw on the map are transcription factors. So these are regulatory pathways where one gene activates another one or represses another one. And this is so-called PRR7 transcription factor. It's within the PRR37 clade of transcription factors, which you can see. Circled is the beet gene with a number of genes from Arabidopsis, rice, and other species. Interestingly, an ortholog has a similar function in barley. You see here, this is barley or cereals in general. A PPD1 gene is a major gene for photoperiod response in barley, and this shares high sequence homology to our floral integrator or bolting time gene BTC1. And they both share homology to PRR7, which has a quite different function. It's a flowering time gene in Arabidopsis, but at a different point of the regulatory pathway. So this is the first gene, and this was cloned from the locus B. And we did haplotype analysis, and we found this is highly conserved among all the cultivated beets. So these are the cultivated beets. These are the white relatives. And they are distinguished by early flowering. Very typical for a wild annual or wild species, it is an annual. And this is a very negative factor in beet breeding. By the way, beet breeding has a history of just 200 years. That means 100 generations. That's not much compared to wheat or rice. 100 generations of breeding. And what happened during these 100 generations? Breeders have selected for just one haplotype at the BTC1 locus. You can see it's completely conserved. All the sugar beets worldwide, and their relatives, by the way, red table beet, an important vegetable, and fodder beet, they are fixed for this haplotype A. So this is a selection footprint left in the beet genome due to 200 years of selection for non-flowering, which is an easy trait to select in the field. You have a flowering plant, you chop it away and discard it and just keep the non-flowering ones. Then we found there's a second major gene. The locus was called B2, and this gene is on chromosome 9. It was cloned by a classical map-based cloning procedure exploiting the reference map. Of course, we're lucky to find a high recombination rate in this region. Used the context from the beet reference map, which has been published some years ago, and found a new gene, which at that time was unknown as a flowering time gene. We called it BvX19 because it shares homology to a gene from Arabidopsis, not known as a flowering time gene at that time. It has 2 functional domains called BB1 domain, and the mutant we used by MapX cloning has a single mutation within the reading frame, which results in a stop. So the protein is truncated and has a different function. And this was used for further analysis. It's important that you see this is a mutant with a stop codon within the sequence, within the reading frame, resulting in a truncated protein. Now, these BvX19 encodes a double B box zinc finger transcription factor. Here you see the clade of different BvX genes sharing homology to constans. And the most famous probably is constans, the integrator of the photoperiod pathway from Arabidopsis. However, Constans has three important domains, the so-called B box and the CCT domains, which are important for DNA binding and protein-protein binding. Our genes, however, is lacking the second domain, the CCT domain. It was interesting because the other gene, BTC1, carries a CCT domain. And by the way, interestingly, we cloned the gene from beet. A few months later came a paper from Arabidopsis where they confirmed it is a flowering time regulator also in Arabidopsis. So a rare case where a gene was first found in the crop and then verified in Arabidopsis. Now, we're interested to know how do these genes interact, because there is no Constans gene in beet. Quite interesting. We find many Constans genes across crops in rice, in wheat, in other crops, but not in beet. There are Constans-like genes, COLs, but no Constans also though. So we were interested in this question, how do they interact, how do they work together? And we knew that they regulate the downstream targets FT. Again, this is the floral repressor. And FT2, this is the floral activator. If this gene is active, the vegetative plant is turned into a generative plant. And we were interested to know how they work together. So we did two kinds of experiments. One is a classical Mendelian segregation in an F2 population. The other were protein-protein interaction studying by yeast to hybrid in vivo fluorescence studies. I will not mention that today. And Nadine Daly, you see the researchers doing the job, who did the job on top right. Nadine Daly, she made a cross between two beets which differ by their haplotypes at the B and the B2 locus. To make it easier, I didn't write BTC1D, just B, D, and B2H. So these are two different haplotypes, H and F, D and A. And this is the mutant I just showed you with the truncated protein. She made an F1, and then we had an expectation. The expectation was that among in the F2 with the 9 different genotypes, there are early bolting ones, annuals, biennials, bolting after vernalization. These are the green ones. And one, we expected, the double recessive class, EA, B2H, which was expected to be never bolting. Why is that interesting? Never bolting means that even after winter, the plant is not able to bolt. You may think this is negative because you cannot produce seeds, but you will see later this is important for breeding. So this was the expectation. What came out? She could confirm that the double recessive or double homozygous for A and each haplotypes or alleles, in fact, is never bolting. So these plants, and this is an experiment in the climate chamber, where plants grew for 325 days, almost a year, after vernalization. So they experienced cold for 10 weeks, simulating winter conditions. And after 325 days, they were not able to bolt. Not any of these plants could bolt. Whereas the normal sugar beets, as you see here, can bolt. These are the controls after vernalization. And the annuals, they bolt very early, even before vernalization. And this is also a sugar beet type, bolting early after vernalization. However, this is not. Interestingly, also two other genotypes, all carrying or all homozygous for the mutated B2 allele, they also displayed some extent of never bolting. Some were bolting, but most of them were never bolting. Now, this was the genetic proof that both genes work together to control bolting. How are they doing that? Then we analyzed the downstream targets. So the first downstream target, maybe I show this again. No, first downstream target is BvFTI, the floral repressor. So what happens in the double homozygous never bolting? The floral repressor in blue, you see, is highly activated, even after vernalization. In a normal beet, the repressor is downregulated after cold, and then the plant can start flowering. Here it is upregulated, clearly indicating that the mutation in these genes results in upregulator of the floral repressor. And the floral activator, FT2, is downregulated. You see, blue FT2, almost no expression in the beets after vernalization. in stark contrast to the normal sugar beet, where this gene is highly upregulated and then the plants are starting flowering. So that was a proof from transcription analysis of the downstream target that both regulate FT, the two FT genes. Now we did a transformation, and in this experiment we knocked down the BTC1 gene by RNAi. So it's an old experiment today, probably using CRISPR-Cas, but RNAi. So we used an annual beet, which is early flowering, and knocked out BTC1. What happens in an annual beet not transformed? The FT1 gene, the floral repressor, is completely downregulated. No transcription, no transcript at all. But if you knock out BTC1, the upstream regulator, the FT1 downstream gene is highly activated. Clearly an effect of BTC1. And more interesting, we found among 10 or so different RNAi plants, or these are offspring of RNAi plants, this is a transcription activity of BTC1. And as you know from RNAi, you never have 100% downregulation of a gene. In this case, there were most of them gray downregulated, not completely, but 2 were still upregulated in comparison to the control. And if we when we looked at bolting after winter, we found those plants where the BTC1 gene was not turned down, they flowered early after winter, the yellow-orange one. While these plants in gray, where the BTC1 gene was knocked out, could not flower at all. Even after winter, they weren't able to flower in correspondence with the transcription analysis, which clearly showed that these plants have a highly activated floral repressor FT1. These are the photos. This is a plant after vernalization, not able to flower. This is an early flowering control. And in this way, we could turn a wild species into a crop. This is a wild relative, which is early flowering, makes no beet, but if we downregulate the BTC1 gene, it is not able to flower anymore, and after 6 months, it makes a huge beet. So with this simple downregulation, we could create a sugar beet, so to say. So how can we use it? Now, I will share with you some models we proposed. So first of all, it became clear that a biennial beet, so a normal sugar beet, under long days, without vernalization, before winter, the 2 genes act together to control downstream targets. So this is BvFTI and BvFTI. BvFTI is repressing the activity of BvFTI. Those 2 are not active, and in the end, there is no floral transition before winter without vernalization. The beet is growing vegetatively. This is a typical culture in the field where the farmer harvests the beet. Now what happens after vernalization, after cold? Again, they work together, now repressing the floral repressor and activating the floral activator, and that results in floral transition and flowering. This is the model we have. Now this is also extended by a model on the protein level, just to show you that we have an idea now how these genes work on the protein level. We know from Arabidopsis the constans gene, which carries the two functional domains, is a repressor of the FT downstream target. It binds to the promoter region of FT, activating FT. In beet, these 2 different genes, BBX and BTC1, with the different domains, form a dimer. And that was confirmed by protein studies. They form a dimer, and they acquire a constans-like function in beet, as in Arabidopsis. And this is after vernalization. FT2, the floral activator, is activated and RNA transcript is produced. But if we use the mutated gene, and by the way, it does not carry the BB2, the second functional domain, those 2 genes, those 2 proteins cannot bind anymore. This was also verified by protein studies. There's no binding anymore. So FT2 is not upregulated, no transcript. This beet cannot flower anymore, even after winter. Why is that interesting to us? We're interested in a winter beet. This is a typical feature of northern crops. A beet which is flowering has no yield anymore. It doesn't even make a real beet. It has a low sucrose content, so it has no value for the farmer. And this typical beet production, it is sown in spring and harvested in autumn. A winter beet, however, will be sown before winter, in August or September, under our conditions. Stay over winter, and next year it would flower. So that cannot be used. This is a beet in the next year, all flowering, cannot be used. However, a real winter beet staying over winter would be sown before winter and would not flower after winter. And now I hope you understand why I showed you the model. We have a model where we can propose non-flowering after winter. We call it never bolting. And this is seen here. This is a beet in the next year, which cannot flower anymore. And this has a 20% higher, 25% higher yield potential. That's quite interesting for production. Now, how can we use the models to breed a beet? How can we use the alleles to design a crossing scheme to produce a winter beet? I don't show you the other models, but I show you how we can use the alleles. And again, this is the mutated allele H in combination with any other. There's dot. After winter, this cannot activate the floral activator. However, the repressor is active, so this beet, even after winter, is not able to flower. And this is a roadmap for breeding a winter beet. By the way, we need many haplotypes, and Nadine Höft in the lab did haplotype studies among beets, cultivated and wild beets from all over the world, and she found some new haplotypes. Altogether, there are 11 BTC1 and a number 7 BvX19, 5, 8 BvFTI, and 4 FT2 haplotypes, which can now be used to cross them to produce the winter beets. We also, and this should be the end of my talk, were interested in quantitative variation. Of course, as you could see from the second or third slide, 150 genes in Arabidopsis, we expect more genes also. We cloned the major flowering time regulators, but there's certainly more genes in beet regulating flowering time. So what we did, we found an interesting accession. This accession, without crossing them with the two BvX and BTC genes, has a low post-winter bolting rate of 0.5. That means even after winter, as you can see here, this is about June, July next year, most of the beets are not able to bolt. And we mapped the region of the genome where this factor is located at, and it's a QTL on chromosome 9, with explaining most of the phenotypic variation. And this QTL was called BR1. So the bolting rates in the F3 are between 0 and 1. So 0 means that none of the F3 could bolt. And this QTL explains 65% of the phenotypic variation. And here you see data from F3 families. This is a marker which is linked. It's within, actually, it's here. It's within the QTL, 2 markers. And those which are homozygous for the non-never bolting allele, they are bolting quite early after winter. This is bolting after winter, while those which carry the allele for never bolting are not able to bolt. And sorry, it's the other way around. This is non-bolting down to non-bolting or rarely bolting, and this is the normal beet, which 100% of the plants are bolting. So the marker BR1, which is located within the QTL, can distinguish the 3 genotypic classes, and bolting is depending on heterozygosity and homozygosity. Those which are homozygous for the recessive QTL allele have a high degree of non-bolting. Again, that's interesting for breeding a winter beet. I also want to show you that Connie Tränkner in the lab did a lot of sequencing, and she cloned, in the end, a candidate gene from this locus, but I want to hurry up and not show these details. This last slide shows now how we can exploit this information for breeding a winter beet. We must have 2 parents because all the beets are hybrids, which are crossed by each other. We can upregulate floral repressors in the parents or downregulate floral activators by RNAi or CRISPR-Cas using mutagenesis. We need to produce seeds, so a never bolting beet cannot be used, unless you can induce bolting, for instance, by chemically induced expression, and then you result in a never bolting beet, which even after winter does not bolt. And this shows a recent paper, a very nice paper from rice, where a Japanese group induced bolting by spraying with an agrochemical. And this is the FT ortholog of rice, which is transcriptionally activated by spraying a so-called agrochemical, so some sort of a fungicide, which is used in rice production. So it's possible to induce flowering in this way. This is my last slide. These are the people we are collaborating, and I would like to give credit to them from the other labs in Germany and Sweden and Belgium. This was mostly funded by the German Research Foundation, and if you're interested in more information, I would like to announce also a meeting. It's a final meeting of a coordinated program in Germany about flowering time regulation, and this will take place in March, and we're happy to welcome you there, and I'll be happy to take questions if there are any. Thank you.
Thank you. We have a couple of minutes for a question. Anybody? I do have one. And for peanut, we concern in the dormancy. So for the FT gene you shown today here, and I spent some time we found some a copy in peanut, but it's still kind of not correlating, highly correlated. What you talk about today, you have another gene, BTC gene, BTC and the FT gene interaction of the flowering. That probably give us that indication. We need to looking for another gene.
Right. Both in dormancy?
Dormancy.
Yeah, well, dormancy may be other factors. I'm not sure about whether FT genes are related with dormancy. It's certainly present in peanut.
It is in peanut.
Yeah. It's certainly present, certainly has been shown, because so far to my knowledge, every crop, every flowering plant carries an FT component. Yes. But I don't know whether it has some impact on dormancy.
Yeah, we did a little bit transcription study, this FT gene expression showing some correlation, but it's not that that high. Anybody? All right, let's move on to next speaker.